Good morning, all. Today, the topic we study is enzyme. Enzyme are a biocatalyst, the catalyst of life. Catalyst is defined as a substance that increases the velocity. What is enzyme? Enzyme is a catalyst which is which increase the velocity of reaction, rate of chemical reaction without itself undergoing any changes in overall process. That is the enzyme. Enzyme may be defined as a biocatalyst synthesized by living cell. They are protein in nature, colloidal and thermolabile in character, and specific in their own action. Enzyme are a biocatalyst, mainly defined as the enzyme are a biocatalyst that speed up a digestion and metabolism. Where is the enzyme located? Enzyme is located in the cells, cytoplasm, mitochondria, tissue, and the body fluid. Then next is the classification of enzyme. Enzyme are named based on the substrate, reaction, synthesize, chemical in nature. On that basis, the classification of the enzyme are based. Firstly, the base on the substrate. Classification is based on the substrate. In that, the enzyme are named by adding a suffix ASE to the name of the substrate. Any substrate which having a name after that we are adding ASE, then the name of the enzyme is created. For example, there are mm, 8 enzymes. For example, carbohydrates. Carbohydrates is when it is the substrate. Then the enzyme name is carbohydrase. Protein is the substrate. Then the enzyme is proteinase. Means which is the name of the substrate. Depend on that the name of the enzyme by adding a ASE. Same as lipids, lipases, nucleic acid, nuclease, maltose, maltase, lactose, lactase, sucrose, sucrase, urea, urease. These are the enzymes which are based on the substrate. Based on the Enzyme Commission of the International Union of Biochemistry, means IUB, enzymes are classified into 6 major classes. First is the oxidoreductase. What is the oxidoreductase? In that the reaction, which reaction is catalyzed oxidation reduction? Transferase. Transferase is the class in which the group of transfer of groups of atoms. Hydrolase in that the hydrolysis is occurred. Lyase in that the addition or remove atoms to form a double bond. Isomerase in which the rearrange of atoms. Ligases in that the combine molecule using a ATP. These are the oxidoreductase, transferases, hydrolases, lyases, and isomerases and ligases. These are the enzymes which is classified into 6 major classes on the basis of enzyme reaction. The chemistry of enzyme is enzyme are specialized large protein molecule made of one or more polypeptide chain. Enzymes may be composed of only protein or combination of a protein and non-protein part. Simple enzymes compose of only a protein. Hollow enzymes or a conjugated comprises of protein and non-protein part, which having a both protein and non-protein part. Protein component called as a apoenzymes. Non-protein part is called as a cofactor. Prosthetic group also there. The prosthetic group cofactor family attached to the enzymes. Coenzymes cofactor loosely attached to the enzymes. Is These are the chemistry of the enzymes, how they attach to enzymes or cofactor factors. Now the properties of enzymes. Enzymes is a proteinase accelerator chemical reaction without itself undergoing the changes. Specific in action undergoes a denaturation, reversibility of enzyme action. Specific in action means that we study in upper slide. Ligases is combine molecules using ATP, or hydrolase is undergoing a reaction hydrolysis. Lactose substrate and the enzyme lactase is act on that. This is the specific action of the enzymes. Mechanism of action. Michels and Menton proposed the hypothesis for enzyme action. According to their hypothesis, the following steps occurred. Combination of enzyme with a substrate attach on the active site of the enzyme to form enzyme substrate complex. This is the first step, breakdown of a substrate. First, in that the combination of enzyme and substrate. Enzyme firstly combines with substrate, then break down the substrate. Active site loosens the chemical bond in the substrate, breaking down the substrate into the products. Means what? When enzyme combines with a substrate, then they act on the substrate. Active sites loosen the chemical bonds of the substrate, and then they are breaked. Dissociation of enzymes. Enzyme finally dissociated from the end product and the free to combine another molecule to the substrate. When first then when the enzymes combines with a substrate, then what they do? Breakdown of the substrate. Firstly, by losing the chemical bonds, then what happen? Then the product is released, and as it is, the enzyme is dissociated from that product and combined to another molecule of the substrate for the dissociation. Formation of enzyme substrate complex. Two types of hypothesis in that: lock and key hypothesis and induced fit hypothesis. In lock and key hypothesis, according to this hypothesis, the active site of enzymes are rigid with proper conformation. The substrate simply fit into the active. This lock and key hypothesis proposed by Emil Fischer in 1914. Look at that picture. This is the lock and key complex. Substrate and active site substrate get fixed into the active site of the enzyme and forms an enzyme substrate complex. Second is the induced fit hypothesis. Induced fit hypothesis proposed by Koshland in 1963. According to this hypothesis, the active sites are flexible. In lock and key hypothesis, there is no flexible active site. This is the active site in the lock and key is rigid and with a proper conformation. But in induced fit hypothesis, the active site is flexible. The enzyme molecule changes shapes as the substrate molecule get closed. Right? Look at this picture. The enzyme molecule changes shapes and substrate molecule get closed. The substrate changes the conformation of the active site of enzyme closely fitting on it. When the enzyme closely fitting on it, then the both are flexible. This is the induced fit hypothesis. Now the factor affecting on enzyme activity. The activity of enzyme affected by the number of factors. temperature, concentration of substrate, pH, presence of activators, presence of inhibitors, and UV light. These are the factors affecting on enzyme activity. When temperature is more, then the enzyme activity is low or enzyme is denatured. Same as in the concentration of substrate is more or less, pH is higher or lower. Same factors are affecting on the enzyme activity. These are we study in the next session. Thank you.